Assalamu alaikum. Bismillahirrahmanirrahim. Today's our topic is glycolysis. As you know about the glycolysis is actually the pathway of oxidation of glucose. This course is the this course is the biochemistry of carbohydrates, and we are studying about the different pathways of glucose metabolism. Glycolysis is one of them. Glycolysis is a very important pathway for the production of energy during the fasting, during starvation, and also during the heavy feed, well-fed stage. In all stages, in all organs, the glycolysis is the one and only process which is used for the synthesis of energy which is needed by the body. So glycolysis have a very vast importance within the body cell. What is glycolysis? First of all, we study what is glycolysis. Glycolysis, as it is shown by the name, it is the glycolytic pathway. It is the glycolytic pathway in which the glucose is breakdown for the production of energy. Glucose is 6-carbon molecule and it is converted into 3-carbon pyruvic acid pyruvate molecules. Glucose is converted into pyruvate by the glycolysis. This is one 6-carbon molecule and it is converted into 3-carbon pyruvate, 2 molecules of 3-carbon pyruvate. What happened in this reaction, in this pathway? This is also called as the Embden-Meyerhof pathway because first time this pathway is discovered by these scientists Embden-Meyerhof, and that's why it is also called as Embden-Meyerhof pathway. These are the scientists which first time discovered this glycolysis. Let's start the glycolytic pathway, glycolysis pathway. In the first step, glucose is converted into glucose 6-phosphate. CH glucose is converted into glucose 6-phosphate 6-phosphate. The glucose is converted into glucose 6-phosphate and the enzyme is either hexokinase or glucokinase. Glucokinase is specific for the conversion of glucose into glucose 6-phosphate and hexokinases can be converted either glucose into glucose 6-phosphate and other hexoses are also converted into phosphorylate. Hexokinase is also present in the any organ in the many organs of the body, but the glucokinase is specific for the liver because its concentration is high in the liver cells. Its concentration is high in the liver cells. ATP is converted into ADP and the 1-phosphate is from the ATP is donated into glucose 6-phosphate. Glycolysis basically consists of two phases. First one is the preparatory phase and second one is the payoff phase. First one is the preparatory phase and second one is the payoff phase. In the first phase, the glucose is ready for the conversion for the production of energy. And in the second phase, payoff phase, the glucose is used for the production of energy. The glycolysis gives the energy. In the second step, this glucose 6-phosphate by the enzyme phosphoglucomutase. Phosphoglucoisomerase converts glucose 6-phosphate into fructose 6-phosphate. Fructose 6-phosphate. So the aldo form is converted into keto form. So the enzyme is the isomerase. Aldo form is converted into keto form. Glucose 6-phosphate is converted into fructose 6-phosphate. Fructose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate. Then this fructose 6-phosphate is converted into fructose 6-phosphate is converted into by the enzyme Fructokinase, phosphofructokinase is converted into fructose 1,6 bisphosphate. Converted into fructose 1,6 bisphosphate. Phosphofructokinase is converted into fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. And in this process, the ATP is converted into ADP, and one phosphate is incorporated into fructose 6 phosphate, and the one phosphate is incorporated into fructose 6 phosphate for the formation of phosphofructose 1,6 bisphosphate. In the next step, fructose 1,6 bisphosphate is splitted up into two molecules by the enzyme aldolase. Dihydroxy acetone phosphate and glyceraldehyde 3-phosphate. Dihydroxy acetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This dihydroxyacetone phosphate is converted into by the enzyme isomerase, because these two are also the aldo and keto forms. The keto form is converted into aldo form and the aldo form is converted into keto form. That's why the isomerase enzyme converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So now these are the two molecules. There are formed the two molecules of glyceraldehyde 3-phosphate. Fructose 1,6-bisphosphate by the enzyme aldolase is converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These two products can be interconverted into each other by the enzyme isomerase. So these both are the keto and aldo forms. So they can be converted into each other by the isomerase enzyme. So the dihydroxyacetone phosphate is again converted into glyceraldehyde 3-phosphate, and now you have the two molecules of glyceraldehyde 3-phosphate. This reaction is the end of the first preparatory phase of glycolysis. Now the payoff stage is started of glycolysis. Glyceraldehyde 3-phosphate, the two molecules now is converted into, the enzyme is dehydrogenase, NAD positive is converted into NADH plus H plus one inorganic phosphate is added to it, and this one is converted into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. In the payoff stage, the first reaction of payoff stage, the glyceraldehyde 3-phosphate is converted into 1,3 bisphosphoglycerate by the enzyme dehydrogenase. From now the onwards, the R molecules are 2 because we have two glyceraldehyde 3-phosphates. Glyceraldehyde 3-phosphate is converted into 1,3 bisphosphoglycerate. Why this is called as bisphosphoglycerate? Because when the phosphate groups are added at two different positions, then the molecule is called as bis, and if they are added at the same position, then they are called as the diphosphates. The inorganic phosphate is incorporated in it. Inorganic phosphate is incorporated in it for the formation of 1,3 bisphosphoglycerate. CH double bond O, COH, H, CH, O, P. Glyceraldehyde 3-phosphate is converted into 1,3 bisphosphoglycerate is converted into 1,3 bisphosphoglycerate. Here is the addition of phosphate group. One three bisphosphoglycerate. Then this 1,3 bisphosphoglycerate phosphoglycerate kinase enzyme. Now ADP is converted into ATP. The enzyme is phosphoglycerate kinase, and 1,3-bis phosphoglycerate is converted into 3-phosphoglycerate. Here from the phosphate group is removed. 3-phosphoglycerate. Phosphoglycerate is converted into 3-phosphoglycerate. The enzyme is phosphoglycerate kinase. This reaction is called as the substrate level phosphorylation. This reaction is called as a substrate level phosphorylation because the substrate 1,3-bis phosphoglycerate provides its phosphate into ADP for the formation of ATP. That's why this reaction is called as substrate level phosphorylation, because the energy is produced and for the production of energy the phosphate group is provided by the substrate. In the next reaction, in the next reaction, three-phosphoglycerate is converted into 2-phosphoglycerate. Phosphoglycerate. Phosphoglycerate, and the enzyme is phosphoglycerate mutase. 3-phosphoglycerate is converted into 2-phosphoglycerate. 2-phosphoglycerate. Why this enzyme is called as phosphoglycerate mutase? Mutase enzyme is used for this reaction because the 3-phosphoglycerate is converted into 2-phosphoglycerate, and the mutase enzymes are acted upon where the reshuffling of the molecule from one position of carbon to the other position. So in this, the 3-carbon phosphate group is instead converted into 2-carbon. Second carbon from the third carbon is moved to 2-carbon, second carbon. So that's why the enzyme is mutase, and the 3-phosphoglycerate is converted into 2-phosphoglycerate. Phosphoglycerate, and then this 2-phosphoglycerate is converted by the enzyme enolase by the removal of one water molecule into phosphoenol pyruvate. Phosphoenol pyruvate is converted into phosphoenol pyruvate. One water molecule from these molecule is removed. Enolase enzyme is actually the hydrolytic enzyme which remove the one water molecule from the 2-phosphoglycerate and converted it into phosphoenol pyruvate. Then this phosphoenol pyruvate in the final step of glycolysis. In the final step of glycolysis, this phosphoenol pyruvate is converted into pyruvate, and the enzyme is pyruvate kinase, and one ADP is converted into ATP. This is also the substrate level phosphorylation reaction. Is converted into pyruvate, and this phosphate group is removed from here and donated to the ATP for the formation of ATP. That's why this reaction is also called as substrate level phosphorylation. This is all about all the reactions of glycolysis. Now we are recall again the steps of glycolysis and calculate the energy production during this cycle. Glycolysis. Glucose is converted into glucose 6-phosphate. Here the ATP is converted into ADP. Then glucose 6-phosphate into fructose 6-phosphate. Then fructose 1,6-bisphosphate. Here is also one ATP is converted into ADP. And then this fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. That is again converted into glyceraldehyde 3-phosphate. So 2 molecules of glyceraldehyde 3-phosphate. And now this glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, and here the NAD positive is converted into NADH plus H plus. Then this one is converted into 3-phosphoglycerate, and here one ADP is converted into ATP. Then this 3-phosphoglycerate is converted into 2-phosphoglycerate. 2-phosphoglycerate is converted into phosphoenol pyruvate. Then this one is converted into pyruvate, and here also the ADP is converted into ATP. So in the overall, 2 ATPs are utilized in the preparatory phase, and 1 NADH molecule and 2 ATPs are formed in the payoff phase. So the net gain is here 2 NADH plus H plus and 2 plus 2, 4 ATPs formed here, 3 into 2, 6. So the total net gain is 10 ATP, from which 2 ATPs are utilized. So net gain is 8 ATPs. The net gain of the glycolysis is 8 ATP. I hope so you have a good idea now about the glycolysis process, the glycolytic pathway of glucose oxidation. If you have any questions regarding these lectures, you can approach me on the WhatsApp group on either on my email address or by putting the comments in the comment section. Thank you so very much students for this lecture. Allah Hafiz.